All right, hello everyone. I've got another thing to talk about as far as our discussions about evolution. I'm out here, I got chased under the carport. It's raining pretty heavily. So I raced in here. I'm out of the rain right now. So, hey, let's get to work. Let's talk about our next topic, which is the origin of species. And so we first need to talk about what is a species, and then we want to talk about how do you get new species. And so think back to Darwin and what got him thinking. He found many unique species on the Galapagos. And so he'd go to a different island and there'd be a similar but different species to other ones on other islands. And so he got to thinking about how do species arise? And so this has been an important question. So this idea of natural selection, it's pretty simple. And when you think about it, it makes perfect sense. And so you can see how organisms can change over time. But how does this ultimately lead to a whole new species? And so how do we get all the different species that we see right now, plus all those different species that you see in the fossil record, all those different species that have ever existed? And so let's go back to our taxonomy. And this is, again, how we organize living things based on their common ancestry. And so you remember, King Philip called out for good soup, right? Domain, kingdom, phylum, class, order, family, genus, species. And so this is a hierarchy and a way we can arrange different living things. And remember that Linnaeus kind of figured this out, and Darwin saw this, that if you study organisms, you can arrange them in these hierarchies. And those that are more, used to be those that were more similar would be grouped into the same groups. And you could arrange things based upon their similarities. And now we realize that many of those similarities are due to shared common ancestry. And so now we arrange things based upon their ancestry, and those that have a recent common ancestor are grouped together. And so of these groupings and of these taxa, the species is the only thing that actually exists in nature. It's the only actual biologic unit. All the others are human-made to try to help just give us some organization to it. But the species is actually a thing. So I can't go out and catch a genus. I can't go out and catch a phylum, right? Well, those are just things that we've made up to help us organize what we see in nature. A species, I can go catch. I can go out and see a species. And so that's why a species is very important. And so we start to ask, well, what makes a species a species? How do we define a species? And the most common way, and the one that we use the most in this class, is the biological species concept. And so if two species cannot interbreed, then they're considered different species. So if two things cannot interbreed and produce viable, fertile offspring, then they are considered to be different species. And so the definition of species is based upon their ability to interbreed. And then, like I said, that's called the biological species concept. And so a species is a group of populations whose members have the potential to interbreed in nature and produce viable, fertile offspring. And so that is a very common way to define a species. So now let's apply that, right? So in this group, are these organisms the same species or different species? Well, do they have the potential to interbreed and produce viable, fertile offspring? They do. So they would all be the same species. Humans are all the same species. Doesn't mean that they actually do interbreed. It's just that they potentially could. Are these two the same species? Could these two interbreed? Yeah, pretty gross. But could they interbreed and produce viable, fertile offspring? No, they cannot. So these two are clearly different species. Are these two the same species or different species? You've got a donkey and a horse. Well, can a donkey and a horse interbreed? Yes, they can interbreed, and they can produce a mule. And so they can breed, they can produce viable offspring, viable meaning an offspring that can live and thrive. But can they produce viable, fertile offspring? No, they cannot. The mule is sterile. And so that's why we consider the donkey and the horse different species. They can interbreed, they can have babies, those babies are viable, mules are viable, but the mules cannot themselves breed. They're sterile. And so that's how we apply the biological species concept to determine if things are different species. So why is the mule sterile? A little side note. It's because of a mismatch of chromosomes. And so if you look at the horse, they've got 62 chromosomes, a donkey, or 64 chromosomes, a donkey has 62. And so when you look at the gametes of each, the horse gametes have 32 chromosomes, that's their haploid number. The haploid number for the donkey is 31. And so when the donkey and the horse gametes come together, you've got 63 chromosomes. And so the mule is viable, but when the mule tries to produce gametes, you've got an odd number of chromosomes. You can't split those chromosomes up equally. You get a mismatch during metaphase of meiosis. And so you can't produce gametes that will work. And so that's what makes the mule sterile. Now, are these two the same species or different species? They look very similar, but they have very different songs. And so a lot of birds use their songs to attract mates, and so in nature, these two are not likely to interbreed because they're using different songs. Now, if you brought them together in a lab, you might be able to get them to produce offspring, and those offspring might be viable and fertile. But that's in an artificial setting you brought them together. In nature, would these two naturally breed together? And they probably wouldn't because they have different songs. So we would call these a different species. And so that's what I'm saying here. Different song, different behavior, so they're unlikely to breed into the wild. So we have that situation where you can take organisms and bring them into the lab and artificially breed them and get viable, fertile offspring. But we don't necessarily consider those the same species because it didn't happen in nature. What about these? Are these the same species or different species? Can you breed a beagle and a lab together? Yes. Will they produce offspring? Yes. Will they produce viable offspring? Yes. The offspring will do great. Will those offspring be able to have babies, be fertile? Yes. So different dog breeds are not considered separate species. They're the same species because you can produce viable, fertile offspring when you bring them together. And in fact, in this particular example, you get about the cutest viable, fertile offspring you're ever going to see. So this is a picture I found of a beagle lab hybrid, a beagle lab, mutt cross. And wow, how cute is that? So all these dog breeds are considered the same species because you can do this. You can cross them together and get viable and fertile offspring. Okay, so once you figure out if something is a unique species, it has to have its own name. You need to be able to identify this species as being separate from other similar species. And you need a name that everyone uses, no matter what language you speak, no matter where you are in the world. We can't have confusion. So we have lots of common names for things. And you use the common name you were brought up with. There's a largemouth bass, but also they call them bigmouth bass. There's a fish called a freshwater drum that some people call a gaspergoo. And so if you're working with these organisms if you're a biologist, you need a way to, you need a name that everybody agrees is the name for this species. And so the technique we use that you've probably heard of before, it's called binomial nomenclature, binomial, two-name nomenclature. And so Linnaeus came up with this idea too. And so the name that we give each species is its genus name, what genus it's in, and then the specific epithet that identifies the species. And these names are Latinized, so you've noticed that they've got a peculiar sound to these names. And that's because we use Latin, because Latin is a quote-unquote dead language. There's very, very, very few people who still speak Latin. Languages evolve. And so there's all kinds of new slang and words take on new meanings, and language changes all the time, right? Say, well, man, that's really cool. Cool used to mean it was just cool. The temperature was low. And now cool is, hey man, it's cool, right? So if people are speaking a language, it changes. And for something like this, for naming species, we don't want the name to change or the meaning to change. So that's why a reason that they use Latin. The names are always italicized. They're always written in italics, or if you can't do italics, they're underlined. This is so you can clearly tell that we're talking about the name of a given species. Now, both names together are the name of the species, not just a specific epithet. So humans are Homo sapiens. We're not just sapiens. We are Homo sapiens. It's, just like your specifically, if I wanted to tell somebody specifically your name, I'd give them your first name and your last name, right? It wouldn't be just John. It would be John Smith or whatever. So both together are the official name of the species. Okay, so that's how you identify a unique species. But again, we have these taxonomic names that help us to organize different species based upon their ancestry. And so for us, for humans, we've got names just like everybody else. We have taxonomy just like everyone else. What kingdom are humans in? Well, of course, we're in the Animalia. We're animals. What phylum are we in? Arthropoda, Annelida, Chordata, or Vertebrata? It's kind of a trick question. A lot of people say, we're vertebrates, right? So we should be in Vertebrata. Vertebrata is actually a subphylum. We're in the Chordata. So organisms that are born with a notochord, a stiff cord running down the back. And then in the Vertebrata, it becomes ossified. It turns into a backbone. So, it was not a fair question. And so we can go down the line and look at the classification of humans. And so our domain, the highest level, is eukarya. And so that means that we've got eukaryotic cells, right? There are different kinds of cells. There's prokaryotic cells and eukaryotic cells. And we have eukaryotic cells. So we're in the domain eukarya. All other organisms, plant, animal, fungi, that have a eukaryotic cell are also in that domain with us. So we have that in common with them. We're in the kingdom Animalia, phylum Chordata, class Mammalia, or mammals, order primates, family Hominidae, and then our genus is Homo, our species name is Homo sapiens. And so then we can compare that to other organisms. And other organisms, and all organisms are grouped by their evolutionary history. And so the closer, the more recent your common ancestor, the more taxonomy you share. And so we can look at chimps. And you see that chimps are in the eukarya. And so, they have eukaryotic cells like we do. They're animals like we are, so they're in the Animalia. They're chordates like we are, same phylum. They're mammals like we are, same class. So there's lots we share with chimps, right? We've got a backbone. We have hair. We're endothermic, homeothermic. We produce milk. These are all mammal characteristics, and so that's why we are in the same class as them. Order primates, we're in the same order, right? We're in the same family, hominidae. Bipedal, or at least more bipedal than other organisms. Changes in the jaw, all these things that identify our family. But they're in the genus Pan, and their species is Pan troglodytes. And so you can tell by looking at these taxonomies side by side that we have a very recent common ancestor, because we have we have lots and lots and lots and lots in common with chimps. Well, why? Because we have a common ancestor, a recent common ancestor, and ancestors pass their traits down to their descendants. And so some of our ancestors' descendants became chimps, and some of our ancestors' descendants became us, and that's why we share so much with them. Now, we can put our taxonomy and compare it to a dog's taxonomy, and you can see that, again, dogs are eukaryotes, they're also animals, they're also chordates, they're also mammals, but they're in a different order, right? And so there's a lot that we have in common with dogs, but then there's a lot that we don't have in common with dogs. And so dogs are in the order Carnivora, family Canidae, genus Canis, species is Canis lupus. And so that means that our ancestor with dogs goes back farther than it does with chimps. Okay, so the biological species concept, the BSC, tells us that things are different species based upon their ability to interbreed and produce viable fertile offspring. So it works pretty well, and sometimes it doesn't work very well. What are some limitations to this? For example, fossils. How do you tell if two fossils could interbreed? Is it asexual reproduction? Asexually reproducing organisms just split, right? They just grow or they bud off. They don't interbreed. Is it hybridization? There's many examples where clear species interbreed and produce viable fertile offspring, but they're definitely different species. Which of these are limitations to the BSC? Well, another trick question, they're all limitations. These are all problems with the biological species concept. And so we have to be aware of them, and this is not a one-size-fits-all definition. So for example, here you've got a fossil of a fish eating another fish. And you see lots of fish fossils. How do you tell if they're different species based upon the biological species concept? I mean, these things lived hundreds of millions of years ago. You can't tell if they were able to interbreed. So that this definition of species kind of breaks down when you look at fossils. Here you've got, some plants that grow by rhizomes, right? Where the root grows off and then a new plant comes up, and the root grows off and a new plant comes up. And it looks like different plants, but they're all the same plant because they're all coming from the same rhizome. They're all reproducing asexually. Well, if this plant reproduces asexually, how do you tell if it can interbreed with another organism? You've got pictures of bacteria splitting here. Bacteria don't interbreed with other bacteria. So how do you test if they're different species? You can't, because they don't interbreed. They're not sexually reproducing. Now, sometimes we have two things that clearly are different species, but yet they can interbreed and produce viable fertile offspring. We call those hybrids. And so here you've got a grizzly bear crossed with a polar bear, and you get a growler bear. But the grizzly bear and the polar bear clearly should be treated as different species, yet they can still produce viable fertile offspring in nature sometimes. So that's a problem, right? So these are kind of limitations. So limitations to the biological species concept. Here's an example of something that I worked with. This is a white crappie, and this is a species of fish. And then I'm holding a black crappie, which is a close relative, but a separate species of fish. This is a hybrid between the two. And so I was able to cross these and produce viable fertile offspring. So should they be considered separate species? Well, I did that in the lab, right? And so if they don't do that in nature, then they should be considered separate species. But they do it in nature sometimes, but they do it in reservoirs, which are human-made, so it's not really natural. So, you know, we still consider these things separate species, but you see why this definition, you know, sometimes gets challenged. You see lots of hybridization in fish, for example. Why? They have external fertilization. And so they release the eggs, they release the sperms, and they can just kind of mix together. And so you get a lot of this mixing sometimes in nature. So because of this, you know, we have other definitions of species. People try to find a better definition of species. And can we come up with a better definition that doesn't have the problems that the biological species concept does? So you have something called the morphological species concept. It defines a species by its structural features. And so you look at, do they look alike? Do they act alike? It applies to both sexual and asexual species. You can apply this to fossils. But you know, these are subjective criteria. You know, they look alike. Well, that's not a good definition. So so this definition has its problems, right? Are these the same species? You know, morphologically, they look identical from this picture, but there are subtle differences. But you know, the morphological species might consider these the same species, but they're not. We know they're not for other reasons. So we've got another definition of species that we try, the phylogenetic species concept, or the evolutionary species concept. It defines a species as the smallest group with a unique evolutionary history. Well, considering that we're trying to, you know, group things based upon their evolutionary history, this is a pretty good definition, and it would apply to any organism, any type of organism, and any species. It's just difficult to measure. You know, it's difficult to measure the unique evolutionary history, but this is so that's a downside to this one. You know, this recognizes that groups with unique genetic composition are important biologically, right? Those unique genetics make them a unique unit and should be treated separately. It should be a separate species. So the bottom line of all this is that, you know, biology's got lots of variability, and it's difficult to get a perfect definition of something like a species, but we have some very good working definitions. You know, the world is shades of gray. It's not black and white. And this is a perfect example of that. Having said that, you know, the biological species concept is very popular, and it's probably the one that we're going to use the most. It'll apply the best in this class. Okay, so now that we've talked about what a species is, let's talk about how do new species form. Where do we get new species from as the result of natural selection? Okay, so we're talking about speciation then, the creation of new species. So we have an idea of what a species is. How do they form? So step one, you have a small population that gets isolated from the main group. So you have a small group of the species that somehow gets split off, isolated from all the other populations. Step two, the gene pool changes in that small population. So the alleles and the relative number of those alleles change in that small population, and they also may or may not change in the original group that the small group split off from. So you have genetic changes in one or both groups, but most likely, you know, genetics can change much quicker in the small group. Step three, over time, those gene pools become so different that the two groups become reproductively isolated. So because they're separated, their gene pools can change over time. And with their gene pools change enough, it's just like with the horse and the donkey. Their genetics have changed so much that when they come back together, then they can't produce viable fertile offspring, and they're reproductively isolated. And so that's kind of what they're trying to show you here in this figure from your book. So you get a population that becomes isolated. These evolutionary changes accumulate over time, And so each population diverges in its genetics. And then you get enough differences that accumulate that they become reproductively isolated. And so these genetic changes, very likely could be natural selection. Could be that one group is adapting to a different environment. But there's lots of other ways that genetics can change too. Genetic drift, assortative mating, founder effects. There's all kinds of things that can go on to change the gene pool of a population. This is kind of a diagram showing what I'm trying to talk about here. These populations, the blue circles represent populations. So you remember, what is a population? A population is in a biology, it's a group of individuals of the same species in the same area. Members of a population actually interbreed and share a lot of their alleles. They share a common gene pool. Members of a species potentially can interbreed, but members of a population actually do interbreed. So the population is a subset of species, right, that actually exchanges alleles. And so, for example, if you look at a map of Kentucky Lake and then right next to it is Barkley Lake, the largemouth bass, that's a fish, the largemouth bass in Kentucky Lake are a population, and the largemouth bass in Lake Barkley are a population, because the bass in Kentucky Lake breed with each other and the bass in Barkley Lake breed with each other. They're the same species because they could potentially interbreed, but they actually only, interbreed within their lakes for the most part. And so each of these circles represents a population where there's lots of interbreeding and sharing of alleles within the population. Now the arrows between these populations represent gene flow. So what is gene flow? Gene flow is when populations occasionally exchange alleles. And so organisms tend to breed within their population and share alleles within their population, but every once in a while an organism leaves and goes to a different population and starts exchanging alleles with that. So it brings some alleles from the first population to the second population. For example, the largemouth bass in Kentucky Lake tend to just breed with others in Kentucky Lake, and the same thing for Lake Barkley. But there's a canal that connects them. So every once in a while, a largemouth might swim from Kentucky to Barkley and then breed with fish in Barkley. So it's taking some of those Kentucky alleles and exchanging them with Barkley. Why can it do this? Because they're the same species. And so this happens, is much more rare than breeding within your population, but that's gene flow. And so gene flow connects the populations together and binds the whole species together. And so all these populations, exchange alleles within their population, but they also exchange a little DNA with other populations. So they can all interbreed because they're not diverging genetically, because they're all kind of sharing alleles. You share a lot more alleles within your population, but you have some gene flow with these other populations, so no population becomes too distinct. And so it remains a group of one species. But what happens if gene flow stops to one of these populations? So this one population is only interbreeding amongst itself. It's not getting any new alleles coming in, and it's not exchanging alleles with other populations. All the other populations are still genetically bound together, and they're still breeding, but this, the one population becomes isolated. When that happens, that population over time can become a new species. That's speciation. So all these other populations that are circled, they're all still exchanging DNA. So none of them gets too different from any of the other ones. So they're all still able to interbreed. If they're able to interbreed and produce viable fertile offspring, they're all still the same species. But this other population, the green population in the lower right corner, has been isolated. And because it's isolated, natural selection is acting upon it a little differently, and you get unique mutations, and you get unique changes in the DNA, and if enough accumulate over time, then those organisms won't be able to interbreed anymore with the other populations. That's a new species. That's how you get a new species. And so that's what we're saying. This population will evolve separately. Given enough time, if it's separate enough time, then it will diverge enough genetically that it can't interbreed. At that point, it's a new species. So this is the basic idea. This is basically how we can get new species. You have a small group that gets isolated for a while, and it changes enough that it can no longer breed with the original group. And so the next question is, well, what stops that gene flow? So that population became genetically isolated. Gene flow stopped to that population. Well, what kind of things can cause that? How does one population become so isolated? There's lots of things can happen, but we kind of have two broad ways that this can occur. And we call one allopatric speciation and one sympatric speciation. Allopatric speciation means in a different place. Sympatric, sim means same. That's in the same place. And so allopatric speciation is when that group is in a different place and physically separated from the original group. Sympatric speciation is when the group is in the same place. It's not geographically separated, but for some other reason it becomes separated. Let's look at some examples. Well, first off, in both populations, in both situations, you've got a small population that stops exchanging DNA with the other populations. It's become genetically isolated. And over time, those populations become genetically different so that they can no longer interbreed. That's how you get a new species. And so that's what I'm trying to show you with this figure here. So you've got a lake. At the top is the original lake. And you see all the fish are orange. They're all the same species. They can all exchange DNA. On the left, you see the water level dropped. And so one population got separated from the other population. So they couldn't exchange DNA, and so they diverge. And over time, they diverge so much that if the water level comes back up, they can't interbreed. That would be allopatric speciation. They were in a different spot when they diverged. On the right, you see the water level didn't change. They're all still in the same lake. They're all still swimming with each other, but something happened to cause one small group to become genetically isolated. And so then it becomes a new species, and that would be sympatric speciation. So examples like here's, you got, if you look at the Grand Canyon, on either edge of the Grand Canyon, you've got two similar squirrels, but they're different species. And so at one point, their ancestors were all in the same population, but as the Grand Canyon started to form, you had these groups that got on either side, and it got harder and harder, and at some point, that canyon got so deep that they couldn't exchange DNA, and so each group diverged, and now you have a separate species on each side of the canyon. Here's another example. If you look on the Isthmus of Panama, on either side you can find tons of sister species. You can find these shrimp, for example. These are these snapping shrimp, which they do it's like cracking your knuckles. They like break bubbles really fast, and it makes a really loud sound. That's how they communicate. They're very cool. But if you look, you can see on one side of the isthmus you have a species, and on the other side you have its sister species, its closely related species. And these species evolved allopatrically. So during the Cretaceous, if you look at the continents, water levels were higher. So North America and South America were not connected. So you had these populations of shrimp that could all interbreed and exchange DNA, had lots of gene flow. And so they were all, you had several species there. But then as the sea levels dropped, the isthmus rose. Now you've got groups that are separated, and they can exchange DNA. And so they genetically diverge. And what you see today is that you've got different species on either side of the isthmus. That's allopatric speciation. And so the groups got physically separated. That's what stopped the gene flow. And so that's very easy to understand, right? Allopatric speciation is like, okay, if there's a small group and they get, a river cuts them off, or an earthquake opens something up, or something happens to physically separate them, geographically separate them, then yeah, they're not going to be able to exchange DNA, and over time, they're going to diverge. What about sympatric speciation? How do you get something that changes, but they still live in the same area? Well, a good example is the salmon. You're familiar with salmon and this whole family of fish called the Salmonidae. If you look at their chromosomes, they have exactly twice as many chromosomes as their nearest relative, the smelts. And so that's probably how they evolved. You had some sort of genetic error. So you had some smelts that were breeding or producing gametes, but there was some sort of genetic error during mitosis or probably meiosis. And so the gametes had too many chromosomes. And it's sort of like, again, you remember when we talked about the horse and the donkey and the chromosome number was messed up? Well, you had this error that occurred with the smelts, and you had a group of smelts that couldn't produce, that produced gametes that had too many chromosomes. So they couldn't breed with other smelts, but they could breed with each other. And so you have this group that now is genetically isolated from the original group, and so you call them a new species. That's sympatric speciation. And so normally during meiosis you produce haploid gametes, but if you have an error that produces diploid gametes, those gametes won't work with the original parental generation, or the other populations around there, because the chromosomes don't match. But they can breed with each other because these new gametes do match. You can breed with your siblings, that all come from the same nest, because you get the same number of chromosomes. You sort of have an instant species. That's one way you can get sympatric speciation. And so with allopatric speciation, the groups are physically separated, and so they diverge genetically. With sympatric speciation, the groups are not physically separated, but they get genetically separated very quickly due to errors in meiosis. So another example, we see a lot of sympatric speciation in plants. Plants, they can reproduce with themselves, they can fertilize themselves, and so it's a lot easier sometimes for them to form a new species. So let me show you an example of how that can happen. So if you look at kale, you're familiar with kale, it's got 18 chromosomes. And turnips have 20 chromosomes, right? And at some point, a long time ago, you had the egg from a kale got fertilized by pollen from a turnip, and it produced a plant that grew. But because they have a mismatch of chromosomes, this plant that grew had 19 chromosomes, and so it could grow, but it could not produce gametes. So it's just like our example earlier with the horse and the donkey, where the chromosomes can come together and you can produce a viable offspring, but the chromosomes are an odd number. And so that organism can't produce gametes because you can't match up the chromosomes. You can't split the chromosomes evenly because you have an odd number of chromosomes. Well, the same thing happened here. You had the kale and the turnip come together. They produced a plant that was viable, that could grow, but because it had 19 chromosomes, it couldn't produce gametes. So it couldn't reproduce with anything else. And so that's why we call kale and turnip separate species. They couldn't produce viable and fertile offspring. But since this was a plant, the plant could still grow. And plants can do things like grow by rhizomes, and the piece of a plant can break off and root. And so plants can stick around even though they're not able, some plants can stick around even though they're not able to reproduce with anything else. Well, at some point, you had an error during meiosis or mitosis. Somewhere you had a genetic error. And that genetic error led to a doubling of chromosomes. And so you had an error during meiosis or something. Somehow you had an event that doubled the chromosomes from 19 to 38. And so now you've got an even number of chromosomes. Now when this happens, we call this a polyploid event, where you change the, not just one or two chromosomes, but you change the entire number of chromosomes. And so when this happened and the chromosomes got doubled, that was a polyploid event. Now all of a sudden, you've got a plant that has an even number of chromosomes that can produce gametes. But it still doesn't have anything it can reproduce with, even though it can produce viable gametes, but it can reproduce with itself. Lots of plants can reproduce with themselves. So now you have a plant that can sexually reproduce with itself and produce offspring, and they can reproduce with their siblings, and then you can get more and more and more of them, and that became a canola plant. And so we can see that this type of speciation from polyploidy, which is similar to what I described with the salmon, that can happen a lot in plants. And when we look at a lot of plants and trace their chromosomes and compare them to other species, we see this. So for example, wheat did the same thing. And so this is an example of sympatric speciation. Lots of plant species are formed this way because it's easy for plants to propagate vegetatively, so they don't have to reproduce sexually. They can reproduce asexually, so you can get a lot more plants in the area, and a lot of plants can fertilize themselves. So when you have this genetic error, you can still fertilize with yourself, but now you've got a bunch of plants that are fertilizing with each other and producing viable fertile offspring that can't reproduce with other plants in the area. You've got a new species, but it formed in the same area as all the other species. That's sympatric speciation. And so, again, in our original example, we've got a population that gets isolated either allopatrically or sympatrically, but it's isolated. Its gene pool changes relative to the other populations, and so once they come back together in contact, they can't interbreed. They're different species. And so over a long time, you can get many unique populations with many unique gene pools, and a very long time, you can get many different species, and that's how we think species can form. Okay, so that's just there's a lot here that we could talk about, but that's a basic idea. You can see how natural selection can genetically change organisms over time. And if a group of those organisms get isolated long enough, they can change so much that they can become a new species. And so again, you have to think about the unimaginable amount of time that this has been going on, and that's how we can get so many different species, and how all living things can be traced back to a common ancestor. So that's speciation. So that's all I have. Let me know if you got any questions, and I'll see you later.